This is a production of Cornell University.
I wanted to start off by just thanking a lot of people. So this slide has the funding sources for this project that I'm going to be talking to you about today. Also, Brian Eschenauer and Betsy Lamb are the two people who actually got me started on this project, which I've really enjoyed the project, so I'm glad that they got me started on this project. We also had help from Jason Dombrowski with the insect diagnostic lab down in Ithaca. He did a lot of insect ID for us and is continuing to do insect ID for us. Paige Munoz is doing bee identification. Brian Brown has done some weed assessments. And then we had field assistance from Tom Ray, Deb, and Ryan. So what you can expect for the next 30-ish minutes, a little bit less than that, I'm going to talk a little bit about the goals for this project. I'll tell you about the treatments that we used, the results that we got, and then also a little bit about grower responses to the project. And that's the extension piece of this. So let's get started with project goals. Because this is the last talk of the afternoon, I am going to ask you to participate a little bit. So I'm just curious, how many of the people who are still on the call have ever visited an actual Christmas tree farm where Christmas trees were growing in the ground? I think that there is an option where you can raise your hand. So go ahead and raise your hand if you have been on an actual Christmas tree farm before. Either no one has been in a Christmas tree farm or the raise hand option does not exist. What about the chat? Can you type in a quick yes or no in the chat feature?
Amara, in the participants window, there are, let's see, a few people who've raised their hands.
Thank you. Let me pull up another window so I can see that. Okay, great. So it looks like about half of you have been on Christmas tree farms. This is good. So here's just two pictures from a Christmas tree farm here in New York. If you haven't been to a Christmas tree farm, one of the ways that it's different from some perennial crops, like for example, fruit trees, is that it's less intensively managed. And at this particular farm, I was pretty excited to notice that there was some nice milkweed blooming, which you can see in the picture on the right. And part of what got this project started was that Christmas tree growers were telling Betsy and Brian, you know, I've been trying to keep some of these flowers around my Christmas trees that I think are attracting beneficial insects like lady beetles, and I think that's helping me with my insect pest control. So we wanted to test this empirically and get some data on this. Because there aren't a huge number of insecticides that are used on Christmas trees, it was also a relatively simple system to start in for conservation biocontrol, where you're minimizing pesticide applications to conserve the natural enemies of pests that are already there. The other interesting thing about a lot of Christmas tree farms in New York, not all of them, is that a big part of their business isn't just growing Christmas trees, it's also agritourism. And so this is a sign from a Christmas tree grower meeting that I went to a few summers ago that a company was selling because they sell these different activities that Christmas tree growers can have around their farm. Often they'll do corn mazes or pumpkin picking in the fall. And so this particular sign I picked because it actually has to do with pollinators. So we were thinking, if these growers are doing some agritourism anyway, there might be some extra benefits to creating habitat that is supporting both natural enemies of pests and also pollinators. All right. So the big picture goal of this project is we wanted to help stakeholders create habitat that supports beneficial arthropods. And there's basically three components to this. The first one is, what is the best way to establish this habitat for beneficial arthropods? And I have best in quotation marks there, because what defines best for you may vary depending on what your operation is like and what your goals are. We also wanted to know which arthropods and how many of them you could attract if you create this habitat for them. And ultimately, if you establish habitat that supports beneficial arthropods, can you get better pest control on Christmas trees? I will warn you that I'm actually not talking at all about this third bullet point today because we have no data on this yet. But long-term, this is a goal that we're looking to address with this project. Before we go any further, I just wanted to get a little bit of information cleared up just in case. So I'm a plant pathologist by training, and I probably didn't know this information before I started this job. So just to make sure we're all on the same page, there's 3 things that beneficial arthropods need. Just like us, we need food, we need shelter, and we need protection from things that hurt us. In the case of beneficial arthropods, pesticides are one of the things that can harm those beneficial arthropods on farms. So in this picture, you see an example of some different flowers. It's helpful if there are flowers that have different shapes, that bloom at different times, so that there's a diversity of resources for the arthropods. And then this is a photo that my coworker Matt Fry took, and I just think it's a really cute bee. A lot of bees are ground nesting, and so they need some space to live, they need some shelter where they won't be disturbed, and also where they won't encounter pesticides. Second thing I want to make sure we're all on the same page with is beneficial arthropods. So when I use the term beneficial arthropods, I am going to be referring to natural enemies of pests, whether they're parasitoids or predators, and also pollinators. So I'm just curious if you recognize any of these beneficial arthropods. You don't have to, if I would say, go ahead and type into the chat box just to keep us all awake here, if you recognize any of these beneficial arthropods on this slide. And if you send it to all the attendees, they should be able to see. Yeah, so people are recognizing especially the lady beetles, ladybugs, lacewings, monarch. Important thing, interesting thing that I learned, this is actually a viceroy butterfly, not a monarch butterfly. And you can tell because it has this black line across the bottom of the wing. Yeah, so some of these are recognizable, some of them are not. Actually, everything on this slide is a beneficial arthropod. And some other time we can talk about each of these in detail. Okay, so next up, I wanted to talk about the treatments. So when I'm saying treatments, what we did was we used different methods to establish this habitat for beneficial arthropods. And this habitat is a mixture of wildflowers and grasses that are going to bloom at different times of the year so that something is always blooming. But there's different ways that you can establish this habitat. So this research was done here in Geneva at Research North. And just to orient you, these are the main FRU buildings, and we're over here on the east end of the farm. And this is the layout for our field. So we planted these wildflower plots on the edges of a Christmas tree field. And each of these green bars represents a row of Christmas trees. And then the purple rows, there's pairs of rows at the right and left side of the field. Those are strips of wildflowers and grasses that are perennial that should be providing habitat for beneficial arthropods. You'll notice they are labeled with different letters because those strips are broken up into different plots with the different methods that we used for establishment. These 4 plots that are in the middle of the field that are labeled H, these are actually one of our control treatments. So this is just, I designated a similar plot area in the middle of the field in the mowed grass row middles between the Christmas trees. All right. So I'm going to go through the different treatments that we use to establish the beneficial arthropods, just to keep you awake and make sure that you are paying attention. I'm not going to stop and read these answers out, but be thinking as I talk through these different treatments, what do you see as the pros and cons to these different methods for establishing habitat? And I know that a lot of you work with particular commodities, so you might think about how that relates to the particular crops that you work on. So go ahead and put your ideas in the chat box as I go through these. I'm not going to stop and read them all out, but hopefully you can kind of follow along with those as I'm going through these slides. So the first method that we used, the first treatment, was to transplant the wildflowers and grasses in the spring. Spring is not the recommended time for transplanting perennials, but it's a time when people often think about planting, so we wanted to include that in our treatment. We transplanted into bare ground. It looks like we used mulch. This is actually just residue from what had been mowed down in the field before we started using it. We were hoping that by not tilling the field before we transplanted, we might reduce the weed pressure, but there was still plenty of weed pressure, which you can see in this middle picture here. So we did have to do some hand weeding because perennials are pretty slow to establish and they need some help out competing the annual weeds. By the end of that first growing season, the wildflowers were looking pretty good. They put on a lot of growth. You can see some echinacea are blooming here. Our second treatment was to do a spring transplant, but to use mulch for weed control. And we're very fortunate that Larry Smart's program had a lot of chipped willow available around the research farms. And so he let us use some of that for our mulch. This I kind of think about as being the Cadillac beneficial arthropod habitat establishment method because it was definitely the most expensive. It takes extra time and costs extra money if you are going to have to buy mulch. We put down about 3 inches of mulch, but it still did not provide perfect weed control. So we still had to do some hand weeding, but I would say it was easier to weed in the mulch and we spent less time doing it. And you can see over here on the right that by the end of that first season, these plots looked really good. Establishing habitat for beneficial arthropods with transplants is pretty expensive. It costs about $2 per plant. And if you have to do a large acreage, it could be really time intensive. So we wanted to look at direct seeding, which is another strategy for establishing these perennial wildflower plots. Again, spring is not the recommended time for direct seeding because these perennials, the seeds often benefit from experiencing the freeze-thaw cycles over the wintertime. Because the plots were pretty small, I did the seeding by hand and I mixed the seed with boiled rice hulls to make it a little bit easier to sprinkle across the surface of the soil. And I raked it in and stomped it in. If you had a larger area, you could mechanize all of this. When you start perennial wildflowers from seed, the recommended weed management strategy is to mow the plots repeatedly. And this will work if you have annual weeds, but not if you have perennial weeds. So the goal is to mow the weeds before they set any seed. So you're basically getting weeds to germinate and then killing them before they can reproduce. This Rudbeckia species, this is Rudbeckia hirta, is one of the fastest establishing of the seeds in our mix. And we did see some of those that first fall. We also wanted to try fall transplanting, and the recommended strategy for establishing these perennial wildflower habitats is to do at least 1 season of weed control and then plant in the fall. So with our fall transplant, we decided to use buckwheat cover crops to smother weeds. We were able to get in 2 crops of buckwheat, and as long as you mow them when they're in full bloom and before they set seed, that kills the plant and you don't have to worry about the buckwheat becoming a weed. Unfortunately, with this treatment, we had to terminate the second buckwheat crop about 2 weeks before our fall transplants came in, and that caused us some issues because the weeds got about a 2-week head start before we got the perennials into the ground. And you'll see how that influenced the establishment of the wildflowers later on. We also wanted to try some fall seeding, which is when you're supposed to plant seeds for perennials. And for these 2 fall seeding methods, we use different weed management strategies. The first one is, the heading says after summer cover crop, which is incorrect. This is after soil solarization. I was a little bit skeptical that soil solarization would work in New York, but Brian Brown actually introduced me to a colleague in Maine who'd had really good success with this. And I thought, well, if it can work in Maine, let's give it a try in New York. So we found some leftover high tunnel plastic. And when the soil was still moist, we covered each plot with the high tunnel plastic and buried the edges really well. And then during the season, if there were any tears in the plastic, we sealed them. Because the goal is to use the clear plastic to heat up the soil. One interesting thing I learned about this that I probably could have looked up, or I'm sure Brian could have told me, was that purslane really thrives in high temperatures. And so we had, in some of the plots where we used solarization, really impressive purslane growth under the plastic, which is what you can see in this picture here. The problem is that that pushes the plastic away from the soil and probably makes it so that the soil doesn't heat up as much, so you get less killing of the young weed seeds of other species and potentially young weed seedlings of other species and potentially some weed seeds. In spite of that, we did achieve maximum soil temperatures of 120 degrees Fahrenheit, and that was about 3 inches deep in the soil in these solarized plots, compared to only 90 degrees in the control plots. And when there wasn't too much purslane, the plots were pretty bare in the fall when we took the plastic off. And then we direct seeded these, similar to what we done with the spring seeding. And then finally, ahead of fall seeding, there was one treatment where I alternated herbicide and tillage. The herbicide to kill weeds, and then the tillage to bring up more weed seed and stimulate germination, and then direct seeded after the final tillage in the fall. There were 2 controls in this trial. So one of them was just to mow whatever weeds were still there. The entire field had gotten some herbicide in the fall prior to starting this project, and then also again in the spring. But whatever grew back after that, we just mowed it periodically. And then FRU had seeded some mixture of orchard grass species between the Christmas trees rows. And so then these were the plots that were right in the middle of the field, and you can kind of see some little baby Christmas trees on the edges here. Okay, so moving on to the results. I'm going to be talking about 2 types of results, first having to do with the success of establishment, and the second having to do with which arthropods we found and how many of them. So we wanted to make sure that we kept track of how much time we spent with each of these establishment methods, because that's a really important thing for growers to be considering if they're going to adopt these methods. And we wanted to be able to tell them, look, this is the kind of input you're going to need if you do this, so that they can make informed decisions. So this graph is showing the time in person hours. So one person working for 1 hour is 1 person hour on the y-axis, and then the treatments are on the x-axis. I'm not showing the mowed grass control in these graphs because it's not really relevant here. It was more relevant for our insect sampling that I'll talk about later. Each bar shows the time that we spent in 2018 in blue and 2019 in orange. And each bar represents the total or the sum of the time for 4 plots for each treatment, because we had 4 replications of each treatment. So it's a total area of 460 square feet, or about 1/100 of an acre. Not surprisingly, if you are direct seeding, it's going to take less time. No one is surprised by that. Doing the extra weed control for that season before you planted does require some more effort. During the first year, definitely the spring transplanting and mulching took the most time because of that extra time spent mulching the plants. But I think what's interesting is that when you look at both years together, those treatments where we didn't mulch, the spring transplant into bare ground and then the fall transplant after buckwheat, are really catching up to the transplant and mulch treatment. And that's because it just takes more time to hand weed when you have worse weed control. So these big orange bars here for 2019 represent the additional time that we spent weeding last year. We also kept track of the costs, either what we actually spent or what we would have spent if we needed to buy things. So again, spring transplanting and mulching is the most expensive because we included the cost of purchasing mulch. All the transplant treatments are more expensive than the direct seeded treatments. And there were some extra costs for things like plastic for solarization. This graph is showing results for how successful our weed control was. And again, this is based on data that Brian Brown collected for us. So the y-axis here is showing the average percent of each plot area that was covered with either weeds in orange or the beneficial habitat plants that we had deliberately planted in blue. The beneficial habitat plants the statistical differences are represented with lowercase letters, and for the weeds, the statistical differences are represented with uppercase letters. And again, these are averages for the 4 replicated plots, and the error bars are 1 standard error above and below the mean. Again, not too surprisingly, when you put in a lot of money and a lot of time, you get comparatively a lot more of a beneficial habitat compared to the weeds, and more beneficial habitat compared to any of the other treatments. And the weed control in the direct seeded plots was not statistically different from the weed control in our control treatment. Hopefully this gets a little bit better over time. The other thing that you can see from this graph is that direct seeding is a much slower way to establish perennials, which no one's surprised by, we already knew that, but it really highlights the fact that yes, you're spending more money for the transplants, but you're getting faster establishment. So I think this gets back to what are the goals, what are the needs? If someone is wanting to establish habitat for beneficial arthropods, how quickly do they need something that looks like habitat, and are they willing to pay for a faster establishment? So I'm just including a couple pictures to show some comparisons of the different treatments. These are the 2 spring transplanted treatments on the left and in the middle, without mulch and with mulch, both looking pretty good. For the fall transplants after buckwheat, it's just taking those plants a lot longer to establish, and I'm still fighting with weeds in those plots, including a couple of weeks ago. So I think this just really hammers home the importance of good weed management. It's been really hard for these plants to catch up from that. The direct seeded plots, these are pictures from July of last year, just still don't look that impressive. The direct seeding method is really a much slower method. It takes a lot longer for it to look anything like this idyllic pollinator meadow that we imagine when, or at least I imagined when I first started this project. The other thing I'll point out on this picture is that these white flowers here are chamomile, which we did not plant, but are also a pretty good resource for beneficial arthropods. So moving on to the insect data, and I am by no means going to show you all of the insect data because it's late on a Friday afternoon. Just briefly, these are the methods that we use to collect insects. Sweep nets are good for collecting insects that are either flying through the air or resting on plants. Pitfall traps are good for collecting insects or other arthropods that are crawling across the ground. And pan traps are good for collecting arthropods that are flying, especially those that are attracted to the colors yellow and blue. And pan traps are often used for sampling bees. The results I'm going to show you, in addition to being not all of the results we collected, are also preliminary. So I'm just gonna put that out there. First, I wanted to talk briefly about the bees. So again, even though this is preliminary data, I think we're seeing some pretty clear patterns in terms of the fact that there's a lot of bees out there other than the ones we think about. So the y-axis here is showing the average number of insects collected over the entire summer. So we had 20 different samplings over the summer. And so for each plot, I summed those and then took the average amongst the 4 replicated plots. The green portion of each bar represents wild bees that were not bumblebees. And then you can see some tiny orange and yellow slivers at the bottom, and those are representing the honeybees or the bumblebees respectively. Most of the bees were in pan traps, and we collected a total of at least 18 different genera, and Paige is still working on the bee ID data for us. Another arthropod I wanted to show you are Carabid beetles. So these are ground-dwelling insects that are great predators. Some of them also eat seeds. They come in lots of different shapes and colors and sizes. Most of these were caught in pitfall traps. And again, I'm showing the average number of insects over the whole summer in the different treatments. We did see a trend here towards there being more Carabid beetles in the treatments that were planted in the fall. So fall transplanting and then fall seeding. That may be an anomaly. We'll have to see how the rest of the data turns out and then how subsequent years of data look as well. Another one of the arthropods we looked at were syrphid flies. Most of these were caught in pan traps and sweep nets. Here's some pictures over here on the right. Some syrphids or hoverflies look like bees, but they're actually flies. They have only 2 wings, which is different from bees. The adults are pollinators, but the larvae are great aphid predators. So here's an aphid that's about to be eaten by a hoverfly larva. Interestingly, we didn't see as many hoverflies in the fields that had the larger plants that were actually flowering more. I don't have a good explanation for this yet. We're going to have to look at this further going forward. So there's a lot more arthropods we collected. If you are curious, I'm happy to talk to you about it some other time, over Zoom probably. I wanted to briefly touch on some of the grower responses we've gotten when we talked to them about these data. So I mentioned earlier that we had some ideas about why this might be something that growers were interested in. And so at 3 Christmas tree grower meetings, we had a total of between 60 and 70 responses for the next 3 questions I'm going to show you. And so we asked them why they would be interested in doing this, what their motivation was. And I was kind of interested to see that the most popular answer was they wanted to protect pollinators. And conversely, thinking that their customers would like it and would be good for their business was not a very popular response. So at least with these particular growers, we were wrong thinking that this might be a good part of their agritourism business. Growers were also very interested in protecting the environment and controlling pests. And then we also wanted to know about what the barriers are to doing this. If you are wanting growers to change their behaviors, I think it's important to understand what is keeping them from changing those behaviors. So the biggest concern growers had was that it would take too much time, which is not at all surprising to me. What was a little bit surprising was that the next most popular response was, I just need more information about how to do this on my farm. I need some more guidance, some more details, and that was actually more popular than concerns about it costing too much money. And then finally, we did ask some questions to measure impacts, at least to measure the intention of growers to change their behavior. So before and after these 3 meetings, we asked growers about the time that they were currently spending on creating habitat for beneficial arthropods, and then what their plans were to do that in the future. So if you kind of draw a curve over the blue bars, which is before the meeting, and compare it to a curve that you draw over the orange bars after the meeting, you can see there is a little bit of a shift, at least in what growers are reporting is their intention to spend time doing this. All right, that is all that I have for you today. If you are interested in this project, I do post about it pretty much all summer long, both on Twitter and on Instagram. And then I also write about it periodically on my blog. So you can follow up with this project on any of those platforms. And if we have a minute or two, I will be happy to answer questions.
Thank you very much, Amara. We do have a couple of minutes. We had, let's see, a comment by Carly. So Carly's curious whether any of the weeds in your habitat plots could potentially be acceptable to have around and benefit arthropod communities. She says, what is a weed can be very context specific, and some may provide some resources to pollinators and natural enemies.
Yes, Carly's absolutely right. And for example, when we're doing the weed assessment, I told Brian, look, if we intended to plant it, count it as a beneficial habitat plant. If we didn't intentionally plant it, count it as a weed. But so that means that some of the things that were counted as weeds were things like dandelions or chamomile or aster weeds, all of which are going to provide pollen and nectar to arthropods. So yeah, there is some, definitely there are some resources and there's some benefits to those weeds. They're not necessarily all bad in this particular context.
Gotcha. One question I had, how long after the meetings did you resurvey the growers? So that kind of green, that orange bar, I'm wondering how much time spent since the before meeting and after the meeting.
Yeah, so it was immediately after the meeting. So like one of the first slides was asking the question, how much time are you currently spending on this? And then right before basically this final question slide was, all right, what are you intending to do in the future? So, I think when you ask people that, it can probably impact their responses. We're also asking about intention, not about what they actually did. So it's an imperfect way to measure change in behavior, but it's a start.
Do you know if they actually followed through with that response? Do you have any other like data that was taken later on?
We have not asked that question yet, but Betsy and Brian have established a pretty good network of Christmas tree growers that they communicate with regularly. So I think that we definitely could ask that question and probably should ask that question. Betsy communicates with them by email all the time. And actually, they've been really responsive on other things. Like sometimes we're working on a grant and we really need to know what Christmas tree growers in New York are doing, and they're pretty good about getting back to her. So we should definitely ask that question.
This has been a production of Cornell University, on the web at cornell.edu.